a new media.
media. I don't know if it's as good, but.
Okay, so I think that we're good to go. So welcome everybody. This is our first seminar for this term, and of course we're going to do everything on Zoom. And before we begin, I have two requests. First, I will ask you to make sure that during the talk you're on mute, so we don't want to hear your dog barking, or that's one thing. And the second, I will ask you to, if you have a question, John's going to keep his chat open, and you can write a question while he speaks. That's one thing. If you have a question at the end during our 15-minute discussion, I ask you to write me, on the chat that you have a question. Don't shout it. Don't say it, and I will approach each and every one and give everybody their turn so they can ask their question when they want to. Okay, so our speaker for today is Professor John Eiler from Caltech. So John is a stable isotope geochemist. He began his career using more traditional stable isotope applications to study igneous petrology, and then moved on with his group to develop the theory and methodology and applications of clumped isotopes for both carbonate and later methane, and developed the clumped isotope thermometry applications, which turned out to be a game changer for the way we reconstruct temperature in geological records. And in recent years, John and his group moved on to to develop and look at increasingly chunkier, more complex organic molecules, this time not looking at single isotopes or even clumping, but trying to really map the entire isotopic structures of these molecules and understand where they come from, okay, through what processes and what we can learn from them about the origins of organic materials in the universe. So I'm very excited to hear your talk, John. Good morning.
Yeah, good. Well, good afternoon to all of you. I know this is the end of your work day, so thanks, Yuri, for your kind introduction. And, it breaks my heart I don't get to be there. I was intending to be in Israel this fall. I thought I'd come spend a couple of weeks and travel around and see friends and give some lectures, different places and so forth. And of course, that's on hold, but I'll be there as soon as I can do it without having a 2% chance of dying. So we'll put that on hold and have to talk like this for now. So like Yuri said, my preference when I give a talk is to have much more dialectical back and forth during the lecture. We obviously can't do that without it degenerating into chaos, but I'll keep my chat window open during the talk. And, I might miss your question because I'm paying attention to my own slides or whatever, but I will try to keep track of things that people are asking as we go. Another thing, I've really sort of trimmed this down. I'm going to try and really just punch right through all the major sort of logical points and background science and things like that. And we're not going to go deep into technical details about exactly how measurements are made and theoretical predictions are done and all that stuff. We'll just blow through that stuff. And if you want to come back and talk about it later, we can do that. But I know people get Zoom fatigue. And it's hard to pay attention for very long. So my wish is that within half an hour, I'm kind of winding up the main message of the talk. Okay, so that's that. Let me quickly get my slides going. I'm keeping open my windows so I see you while you see me, because I find it extremely disconcerting to be giving a talk to myself or my computer screen or something like that. So if you are having some kind of visceral reaction to something I say, you should feel free to make a grimace, laugh, jump up and down, whatever you want. Okay, so actually now I see I'm not really seeing my chat window. Well, that's too bad. So you'll just have to wait, I guess. So the subject today, we're going to talk about prebiotic chemistry. That's the chemistry that enabled the emergence of life, that basically bridged the gap between the ubiquitous volatile chemical compounds that we know are sort of everywhere in geological and cosmological settings, to the chemistry of life, which involves the same elements. It often interacts with the same common compounds, but of course it does so through chemical mechanisms and using chemical compounds that are wildly more diverse and much more complex in structure than anything that we commonly see in non-biological natural systems. So it's a first order question. How in the world does that happen? It's a chemical question, it's a biological question, it's an Earth history question, it's a cosmological question. It really is a major problem, and it's been a difficult one to wrestle with for reasons that we'll discuss. So there's many different ways that you can approach this problem. My group is trying to forge a sort of unusual new way of doing this, of bringing together very high dimensionality, that is, lots of independent constraint observations from isotope chemistry to deconstruct the chemistry that was responsible for the formation of prebiotic compounds in natural environments. So also you're going to hear, like, I have 4 dogs and they're not well behaved, and so you're going to hear like lots of barking and who knows what else they might do. Okay, so there can't be a better moment to give a talk like this. Hopefully some of you are watching your YouTube video streams or something in the middle of the night for you. 14 hours ago, the OSIRIS-REx mission touched down on the asteroid Bennu and attempted to collect something like a few grams to maybe 100 grams of solid rocky and sandy material from the asteroidal surface. So they've been preparing for this for multiple years. They've probably been, at least thinking about doing this mission for over a decade. But this spacecraft has been in orbit around the asteroid Bennu, shown here for scale with the Israeli Sarona Tower. First, for a familiar reference, I had to look that up. What's the tallest building in Israel? And apparently that's it. Also, if you have not been to visit that in Tel Aviv recently, there's the Eiffel Tower, which some of you will also recognize. So they've been in orbit around this body, this like 500-meter diameter body, for like a year, looking, mapping, rehearsing the act of closely approaching it and then dropping a sampling probe and then backing away. And then finally last night, they did it. And they don't know what they collected, or they haven't said what they collected, maybe nothing, because it's not that easy, but they tried, okay? And so this is one of the major motivations for this mission. Yes, the question is why? They spent probably $1 billion to do this, 1,000 people, spent a decade. Why did they do this? What is the point of doing this? And there are a number of different motivations. You'll see lots of different scientific inquiries that are focused on these samples. Even if the samples don't come back, there will be a lot of science done on just the imaging and remote sensing geophysics of this body. But really, the reason they did it, the reason they spent the money, the reason they spent the time, was to ask questions about prebiotic chemistry and the origins of life. And this is part of a broader context of increasing intensity of focus on these questions. First, identification. Can we recognize the products of life in other environments? Some of you may have seen this crazy paper that came out a week or two ago about phosphine in the atmosphere of Venus, and that being a biological product. So that's part of this problem. They may be interested in reconstructing processes of prebiotic chemistry or biochemistry. If I have a material and I recognize it as either prebiotic, that is non-biological, or biotic, something made by life, now that's just the beginning. Now I have further questions. How was it made? From what was it made? And so forth. And then finally, there'll be questions of habitability. Can I take a material from the environment and recognize whether it was formed in a setting where life is possible? So this might be a good example might be materials that will return from the surface of Mars. We'll want to identify whether the organics, for sure, we'll find organic molecules. There are organic molecules everywhere in the universe. There's organic molecules on the surface of the moon. So we will get organic molecules back from Mars. We'll eventually get organic molecules back from the icy moons of the outer solar system. But then we have to start asking questions. Are they biological? How were they made? Did they come from environments where life could exist? So this is really the motivation for the OSIRIS-REx mission to Bennu. And here are some upcoming, some simultaneous and upcoming events. The Hayabusa 2 mission is also, it's a lot like the OSIRIS-REx mission. It's run by the Japanese Space Agency with various partners. And it has gone to another asteroid with a funny name, another of these undifferentiated primitive bodies. They're basically the sedimentary rocks of the early solar system, and has collected some dust and is bringing it back. It's supposed to be here, I think, in December, something like that. And similar goals to the OSIRIS-REx mission. Of course, everybody knows about Mars 2020. The intention is to bring back sedimentary rock, to bring back organic bearing materials, and then ask exactly these same questions. And then there aren't any clearly identified sample return missions yet, but there's a lot of work going on behind the scenes and trying to decide how will we do this in the outer solar system? How will we return materials from Enceladus, from Europa, to terrestrial laboratories to search for evidence of life or of prebiotic chemistry? So there's a lot of different scientific approaches to this question of life's origins. And the problem is that we have absolutely no record and no clearly defined, testable, falsifiable hypotheses about the critical moment when non-life becomes life. Outside of a Frankenstein movie or something, or a myth, you really don't know exactly how that happened. And even people who've spent their lifetime studying it can't articulate exactly what they mean about how it happened. So there's always this veil of vagueness, of mystery about the problem. But the further you get away from it, the more concrete it becomes. It's like I have to wear these crazy glasses now to see my computer screen. The further away I get from things, the easier it is for me to know how to ask questions about it. And so one way to get far away from this critical moment is the top-down approach, where you back way off and ask questions about the properties of planets, the properties of potential ecosystems, the habitability, the lifestyles, the ecology of large integrated organisms. That's one way to do it. But of course, you'll come to the point where you have to say, okay, now I've described the smallest little microbe, then what? Okay, how did I get there? The other approach, which is the one we're going to follow, is to go to the other end of the spectrum, to start with the smallest and simplest chemical compounds and ask yourself, how did I build up those compounds into simple organics, methanol, formaldehyde, and so forth, organics that are used actively by life in really critical ways, like amino acids, organics that just like don't seem to be possible to exist outside of life, like RNA, and then somehow all of that gets integrated into something that is alive. So building up from the bottom or deconstructing from the top. These are the two approaches people take. We're only going to focus on building up from the bottom, on the chemistry side to this problem. So the chemistry problem it's complicated because so many different things have to go right in so many different ways in order to have non-biological organic chemistry come together to meet the functional requirements of life. So one of them is you need structure and confinement. So somehow you have to go from precursors through some kind of polar compound, almost certainly like a fatty acid, lipids, which are more complicated structures built up of fatty acids, and then somehow assemble them into cell walls. That has to happen. We need information that can be copied faithfully, but with small variants. So we use DNA, obviously, for this. There's other ways you could do it. That function could be met in other ways, but basically you need to go from simple precursors. All of the ways people have thought of involve making nucleobases, which we'll come back to later. These have to somehow polymerize or oligomerize more properly the larger structures. Then they have to be able to replicate and amplify to sort of build up structural versions of. Need to be able to manage an energy metabolism. So you need to be able to go from precursors through simple organics like aldehydes and so forth to amino acids and sugars, and then big complicated things that are good at shuttling energy from one chemical reaction to another, like NADP. We have to consider the chemical kinetics. So initially, you are just beholden to the rate laws of abiological reactions, maybe with some special catalysts like surfaces. But eventually, if you're a living thing, you want to take control of that. You want enzymatic activity so that you get to decide the kinetics of where and when reactions occur. And then, whoops, that's enough. Apparently that's enough. I thought there was going to be another one, but that's only 5 things. You only need 5 things. Okay, so that's pretty complicated. There's a lot of different things to think about there. Let's break it down into a simpler sort of approach. There's really 2 major steps that need to be addressed, and they just happen to be happening simultaneously with different classes of compounds. One of them is you have to be able to take ubiquitous substrates, methane and hydrogen cyanide and so on and so forth, ammonia, and things that you can easily make in many different environments. And they have to undergo a chemical synthesis to make what I'll call enabling organics, organic molecules that they are not life. You can make them lots of different ways without the activity of life, but if you don't have them, you're not going to make anything we would recognize as life. You might make some fanciful idea of life with silica or made of diamond crystals or who knows. That's fine. You're off having your own fantasy there. If it's going to be life like we understand how it operates, you need to first make these sorts of enabling organic molecules. And then those compounds will undergo oligomerization. They will join to one another. They will undergo organization at the nanoscale, the microscale, the milliscale, macro scale. They will organize themselves into functioning operational systems. So a lot of the people interested in this focus on this second half for good reasons. This is how you go from not alive to alive. But it's also a problem that most of those processes and most of those structures, they're not well preserved in the geological record or the planetary materials record. And so if you feel like your job is to learn not what is possible, but what actually happened, then you're going to have a hard time even beginning with that. This other part of it, the initial synthesis of enabling organics, that's potentially solvable because we find these enabling organics in many different geological and planetary environments, and it is imaginable that we could figure out how they formed. So that's what I'm focused on. So a lot of this field is focused on what I refer to as scenarios. They're not really hypothesis testing so much as hypothesis building. They will have a concept in their mind of how the origin of life likely operated. And because there isn't an easy way to directly observe how that occurred, and there aren't easily interpreted records, they will go into the laboratory or theory or just conceptual models to build up an idea. And the richer the idea gets and the more real it feels, the stronger it is. Okay. And sometimes the idea will encounter a totally insurmountable obstacle, but the history of this field is generally not. Generally, you get to keep any idea you can come up with, you get to develop. And so we have these parallel magisteria of competing concepts, scenarios. An old one is the primordial soup. You have an ocean full of reactive organic things. This isn't very popular anymore because of its dilute chemistry. Hydrothermal vents, you create a special interface between hydrothermal fluids and the ocean. You do your chemistry there. Lakes, that's another interface between water and atmosphere. It's another place where you were able to concentrate critical reagents and bring together reagents with different environments of origin. Clay surfaces, this is to take the same concept that you need to localize the chemistry and really refines it, brings it down to a tiny, tiny scale to imagine that naturally occurring clays can act as catalysts and reservoirs. They're basically little microcosms that are able to do sort of like natural chemistry on a chip. Subsurface fluids, this is another place where you can have gradients in critical chemicals that sort of serve the same purpose as these interfaces, bringing together compounds with origins in different environments where they can react. And then another idea, panspermia, it basically says, I don't know what the origin of life is. It happened somewhere else and it came here. It fell out of the sky. It's not a very satisfying answer, but it actually is one that you'll see a surprisingly large number of papers written about. Okay, so two of the major challenges are you find a molecule and now your job is assign it to one of these origins. How are you supposed to do that? And then how do you know the difference between a molecule that was forged in one of these natural settings versus a molecule that was produced by life? So let's now talk about some of the one level deeper detail. We'll pull out a couple of concepts that we just discussed in generalities and talk about some of the specific problems and focus on ideas that are more prevalent right now, things that people are working on in a more focused way. So first of all, a little context. It's not easy to make a carbon-carbon bond. That's the basic problem. There's lots of ways to do carbon chemistry without the activity of life. This is everywhere. This is in all geological environments, all planetary environments, cosmological environments. You're doing carbon chemistry, but it's not very easy to make carbon-carbon bonds, and it's particularly not very easy to make elaborate organic structures. So in the cross-section of the Earth, or any other sort of similar terrestrial planet, the three kinds of chemistry that we know are active in some of these environments almost ubiquitously are redox chemistry, interconversion of oxidized and reduced forms, like turning CO2 to methane. Polymerization, I can take two methanes and make an ethane and a little hydrogen, and maybe I do this one or two more times. That's possible. And then acid formation, so making simple organic acids, like by reacting methane and CO2. This is also possible. The problem is, thermodynamically, the energetics of the redox chemistry only favor reduced organic carbon compounds like methane at extremely high pressure, meaning deeper than about 200 kilometers in the Earth, higher pressure than any pressure that can be achieved in primitive bodies like asteroids and small moons. So it's pretty difficult just to make a reduced carbon compound in these settings. And then the polymerization chemistry and organic acid forming chemistry is both inefficient and only occurs at high pressure in planetary interiors. If we focus in the upper part of planetary interiors, the upper few kilometers, where you have magmatic activity, the interaction of a hydrosphere maybe with the crust through hydrothermal circulation, now you have a little more possibilities. The magmatic systems and their volcanic output, these remain hopeless, okay? At low pressure and high temperature, you simply don't begin organic chemistry at all. But in the hydrothermal setting, particularly below 300 degrees C, several things are happening. You are able to make inorganic acids like bicarbonate, carbonic acid. You're making a reducing compound H2 by water rock reactions, serpentization. Methane becomes stable with respect to other compounds below about 300 degrees C. So you start to be able to do a little bit of organic chemistry. And we know of mechanisms for making simple organic acids. They can become thermodynamically stable again at low temperature. But that's about the limit of it. Many of the other things that we talked about that we want, we can't have yet. So people have turned to non-equilibrium chemical reaction processes as ways to address this hurdle, ways to get over this energetic barrier that equilibrium thermodynamics doesn't want to do the critical prebiotic chemistry for us. And one of the more frequently cited chemistries is the Fischer-Tropsch chemistry, which is an industrial chemistry made developed to create synthetic gasoline, basically. And the way that it works in industrial settings is you take carbon monoxide and molecular hydrogen and expose it to a catalytic surface where you can really play with the electronic structures of the carbon and hydrogen that are stuck on that surface. They can freely gain and lose and rearrange their bonding orbital electrons when they're in contact with metal. And through various bond rearrangements, they can convert the carbon oxygen bond into a CH bond, and they can even polymerize and form extended hydrocarbon structures. You can make some simple organic acids this way. You can make lots of different hydrocarbons this way. It requires reducing conditions, source of carbon monoxide or CO2, and the main thing is it needs an effective catalyst. It's readily achieved in high temperatures in the lab with metal catalysts. Natural examples, there are a few natural examples of this occurring that seem like pretty strong cases. Most of them not. Most of them are pretty speculative, where people have identified Fischer-Tropsch chemistry. It's not clear that it works very efficiently in nature. Another important kind of chemistry we need to think about as feeding into prebiotic chemistry is catagenesis, that is the breakdown of big extended organic networks. And in geological settings, the way this works is you bury biomass, so lignin and sugars and amino acids and so forth, and they oligomerize into human, so a sort of gunk, and make organic acids. And then through further polymerization, condensation reactions, ring-forming reactions, and then eventually cracking or bond cleavage, thermally activated bond cleavage, they will start to break down and liberate free organic molecules of various sorts, mostly hydrocarbons or organic acids. So an immediate reaction to this should be, well, this is not a very good way of doing prebiotic chemistry because it's a chicken and egg problem. Okay, you're going to make your prebiotic chemicals from something that originally came from biomass. So how did you get started? But that isn't totally fair because there are a lot of cosmochemical settings where kerogen-like materials, big extended polycyclic aromatic hydrocarbons, graphitic sort of particles can form almost like a soot. And then those materials you could imagine undergoing catagenesis in a planetary interior and releasing things of prebiotic value, if you will. I would say the most well-developed chemistry scenario is something called the cyanosulfidic hypothesis. And it is mostly focused on lakes as an environment of prebiotic chemistry, but it could work in a few other settings. The famous Miller-Urey experiment is a sort of uncontrolled, semi-understood version of the cyanosulfidic chemistry. Now people tend to focus more on really building up very well understood laboratory reactions into networks. And here's an example of one of them from the Sutherland group. So in this case, you take your ubiquitous volatiles, and it's really critical that you have cyanide among them, and then and ammonia, and then you do amination, nitrile formation, which is a cyanide chemistry, hydrolysis, reaction with water to sort of oxidize carbon sites. And that is how you create your amino acids and your sugars and your lipids and so forth. So this is a pretty popular idea. And then finally, there's a new paper out just a few weeks ago by Wolos et al. It's an interesting example of how modern computational tools are enabling people to basically take their hand off the steering wheel and say, I'm not going to make up a scheme. I'm going to let the computer make up a scheme. So what I will do is feed it a few molecules and I will tell it, do whatever you want, permitting certain energetic restrictions on the free energies of formation and the maybe activation energies when they know them. It's mostly an equilibrium or quasi-metastable equilibrium chemistry sort of scheme, but sometimes it will detect ways of doing the chemistry that are that you would not have thought of. And so this is an emerging way that this is done. I'm not going to go into any of the details here. In broad brush strokes, the thing it returns to you looks a lot like the cyanosulfidic hypothesis. When you seed the models with cyanide, it uses it to build things. Okay, so where does Bennu fit into all of this thinking? It serves four different purposes. The samples from it will presumably let us describe the compositions of organics that can be made in early solar system settings and the formation pathways for making prebiotically interesting molecules that could have been delivered to habitable planetary environments. Like maybe Bennu was a little sandwich that was being delivered into a habitable setting, and then its chemicals were feedstocks for life. That's when you think about the fluxes of these objects and the fluxes of chemicals needed to support life, that's not super plausible, but it is one thought. Another is it's not necessarily the material of early life, but it is a model for how the habitable interiors of primitive bodies in the outer solar system could have worked. Okay, like the interior of Enceladus, maybe the chemistry there works sort of like the chemistry that operated in Bennu. Another useful thing of it is to say, you know, maybe I'm not that interested in the prebiotic chemistry of this particular setting, these primitive objects, but I will learn about the natural products chemistry that is possible outside of the hood in my laboratory, outside of my computer. What does nature do when it is presented with these feedstocks of cyanide and things like that? How does it work? And then I'll take that and that will inform my models of prebiotic chemistry in more interesting settings like lakes and oceans and things like that. And then finally, another reason to go to Bennu is, you know, it's a natural object of its own value. Like even if you don't care about prebiotic chemistry, you should be interested in the early solar system. You should be interested in asteroids. And so it has its own merit to understand it. So those samples are not here, and maybe they never will be, okay? But we have other objects that are basically accidental samples of very similar objects. So here's an example of the organic compounds that are found in the Murchison meteorite. And we focused mostly on this because it's been so well studied by previous groups. It is a chunk of a primitive asteroid that fell to the Earth as a meteorite, and it contains in it a lot of carbon. So most of the reduced carbon is as macromolecular material. That's basically like kerogen, like the kerogenous material of the early solar system. And then it has a fair amount of carbonate as well. But then in the trace constituents, there's all kinds of crazy things. There's amino acids, there's alcohols, there's aldehydes and ketones, there's sugars, there's ammonia, and so on and so forth. And when you consider that many of these constituents are highly volatile and left the rock soon after it entered the Earth's atmosphere, you should imagine this is just the relatively easily preserved residue of what was there. There may have been a lot more of the highly volatile things like formaldehyde and ammonia and so forth. So this rock, basically, it lets us practice for studying rocks returned from Bennu. It also lets us learn about all of these questions that we're interested in without having to go up into space, which I don't want to do. I'm not interested. Some scientists will tell you, oh my God, I wish I could be an astronaut and go into space. Not me. I don't want to go to space. Okay, so here's the basic challenge. We have our reaction networks that are responsible for prebiotic chemistry, and they're inhabited by sugars and lipids and nucleobases and amino acids and so forth. Well, these are all exactly the same compounds that we find in the central metabolisms of extant life. Not just similar, they're the same compounds. And so how is it that we are just recognizing the molecule, even recognizing the mixture of molecules is not helping you? You need to recognize how it was made. What was the synthesis pathway? It's sort of like if somebody handed you a pile of trains and said, train cars, and said, I want you to tell me, is this from the subway in London or New York? If you're not allowed to look very closely, it might be hard to say. You don't want to see the train cars. You want to know what the map looked like. How did the trains move underground? And if you saw the map, then you know whether it's New York or London. Okay, so we need the map. How were these things interconnected? Okay, so isotopic composition helps us do this in a number of different ways. One is inheritance. So if I am able to use isotopic content like the carbon-13 proportion or deuterium proportion or whatever as a kind of label. I'm hearing somebody. Okay. So I have a label for each of these atomic sites. I can trace it into its products. That's one obvious way. This is how labeling experiments work, and because natural atomic positions in molecules differ in their isotopic content, everything is sort of naturally labeled. So you could imagine using isotopes to do that. Another way that you can reconstruct reaction networks is through the study of chemical kinetics. Basically, I'm not just transferring the rare isotopes from the parent molecule to the product molecule, I'm also, the rate of reactions is a function of the isotopic content of the parent. And so I'm modulating, I'm changing the isotopic content of the product as a function of the mechanism of the reaction. And if I understand the rules by which this operates, if I know that for a particular kind of chemistry, I will change the isotopic content of the product relative to its parental site by a certain amount. If I know that rule, then I can recognize the mechanism. There's also equilibration. A lot of chemistry, including a lot of biochemistry, is reversible. And when it's reversible, it will garden its way to a thermodynamic equilibrium. And that will lead to very predictable partitioning, separation of isotopes between between coexisting molecules in an identifiable way. And then finally, mass transport processes operate differently for light and heavy isotopes. And typically, a heavy thing moves slower than a light thing. And so that also is a way of using isotopic identity to recognize process. So how have these concepts been used before to interpret the organic chemistry of the Murchison meteorite and other primitive organic rich materials? Here's a plot of the carbon isotope content on the delta notation, the PDB scale. We don't have to really worry about nomenclature so much. If it's high up, there's lots of the rare isotope C13. If it's low down, there's not so much of it. And then here, this is plotted versus the number of carbons in the molecule, as if that were an organizing concept for the chemistry, bigger, smaller. And what's being noted here is that all the really 13C rich materials, which are really quite unusual, terrestrial organic matter is typically down here at about -20 or -30. All of these noticeably 13C rich extraterrestrial compounds are small. And the bigger ones look more like terrestrial components, okay? One interpretation of that is that it's a result of the Fischer-Tropsch type chemistry, where I start with something small, and then I have kinetic limitations on my rates of oligomerization, of adding carbon-carbon bonds, so that the bigger I grow, the more I select against carbon-13. So that's one interpretation. Another interpretation is that there was something big and 13C poor in the precursors, and something small and 13C rich in the precursors. And you're seeing a mixing relationship between these two, that the chemistry, it's basically labeling, that I'm seeing a label of two different end members. Another thought is maybe some of these trends that are inverse, where the smaller you get, the poorer you get in 13C, that's evidence of catagenesis happening on asteroids, that you begin with some moderate 13C content, and then the rate at which you break bonds is slowed by 13C presence. And so the things that you make by breaking the catagenetic products, they are 13C poor. That's another idea people have had. And then some people think some of this is just terrestrial contaminants. It's just, fingerprints or whatever. So these are all plausible, and it's not an exhaustive list. The problem is they're all kind of vague. They're not really chemical reactions. They're just suggested loose families of processes. And it's hard to tell them apart. How do you know which is right and which is wrong? So the way we're approaching this is through studying isotopic structure. And the reason we're doing it is it offers more independent constraints, more dimensions of variability to let us interrogate. So first of all, there's the compound-specific composition. That's the kind of data we saw before, but I would say interpreted in a kind of simple way. Then there are site-specific differences. What are the differences in isotopic content between non-equivalent positions in the molecule? Then there are the multiple substitutions, the molecules that have two rare isotopes that are stuck to each other, or perhaps multiple rare isotopes that are not stuck to each other. And when you go through, and you can do this for all the different elements, carbon, nitrogen, hydrogen, everything that has multiple isotopes, When you work through all the combinatorial math of this family of substitutions, even something very simple like this amino acid alanine has over 4,000 isotopic forms. And in some sense, they are all unique. Okay, every one of them is its own chemical compound with its own probability of being made and its own chemical kinetics and its own physical properties like moment of inertia and diffusion coefficient and so forth. They're all unique. They're all independent votes on the chemistry. The technologies that we have today allow us to observe about 100 of them. We haven't ever observed 100 forms of alanine, but we're not that shy of that. We can observe dozens of forms of it. And most of them are multiply substituted. So that gives you a sense that this idea of clumped isotope geochemistry will be very important for the study of organic molecules because that's where most of the information is. So the way that we're using isotopic structure is through a sort of virtuous logical loop where we first of all formulate a coherent chemical hypothesis. So where it's not just, we don't just say, oh, I think it's Fischer-Tropsch chemistry, or maybe it's mixing of something with something else. No, it's a specific family of elementary chemical reactions. So often we have to overreach and say, I'm going to postulate something that I don't have very strong evidence for right now, but I have a little evidence for, and I'm going to be concrete about it. I'll be very specific about how the chemistry operated, and then I will inform that hypothesis with experimental and theoretical constraints on how the isotopic chemistry should work, and I'll predict the isotopic structures that should result from that specific hypothesis. Then I'll go and measure a sample and observe some diagnostic property that I think is going to matter to me. And then depending upon what I see, I'll either go, well, that was a stupid idea, or I'll revise it a little, I'll tweak it and say, well, maybe I should have done a little more of this reaction, a little less of that reaction. So this isn't a technology talk. The last time I spoke at Hebrew University, I talked quite a bit about the orbit trap and so forth, when it was just coming online at that point. I'll just mention there's a lot of different technologies that come into play for making these sorts of measurements. Most of it is mass spectrometry, where we break the molecule up in the ion source or by collision and then study the isotopic contents of fragments as a way of telling what the structure was. There are complementary technologies involving spectroscopy, either NMR or infrared spectroscopy. They only work for a few compounds and special sample types and sizes, but they're very useful as complements to mass spectrometry. Okay, so here is the first major thing that we discovered in this approach, which is that the carbon-13 enrichment that's characteristic of meteoric organic. So notice one of the things I said to you is terrestrial organic matter is typically down here, sort of -20, -30 per mil on this scale. And a lot of the juicy, interesting prebiotic compounds in meteorites are higher than that, tens of per mil higher than that. So why is that? In the case of alanine, at least, the reason is a single site, okay? Two ends of the molecule, the carboxyl end and the methyl end, are totally normal in carbon isotope content. They're indistinguishable from terrestrial amino acids. It is only the amine carbon site that is 13C rich, and it has had its doors blown off. It is 150 per mil higher than the other sites in this molecule, than terrestrial organic matter. This is much larger than common kinetic isotope effects. You could come up with some special scenario in like ice grains at 10 Kelvin or something like that, where you could make such a big kinetic isotope effect, but it's really very noticeable. Something special explains this. And by the way, it's also unquestionably extraterrestrial. So we wanted to develop the hypothesis that this molecule was forged by a cyanide chemistry called the Strecker chemistry, and that the reason why this amine carbon site is high in 13C is that it inherited that signature from an interstellar medium precursor. So the first step in developing that hypothesis is study the Strecker chemistry in the laboratory and by theory, and try and come to an understanding of how that chemistry modulates the isotopic content of the different sites, so that we could look at the molecule and then predict what the precursor's isotopic content was. So this is a synopsis of this, where we took an aldehyde, acid aldehyde, we reacted it with cyanide and ammonia, we make various intermediates, and then the amino acid. And when we do that, the our group, the methyl group, is just passed through without modulation because no chemistry happens at that site. The amine carbon and the carboxyl carbon are lower in C13 than their precursors by some specific amount. And now we can take that as a kind of transfer function and go and make some statement about what we think the precursor should have been like. So this then is the hypothesized network for making alanine. And we can populate that with all of the carbon isotope contents and fractionations that need to be true for this to all hang together. So this is what the precursor has to be like, delta 13C of 150 per mil. And then it has to have another carbon pool in the precursor that can make the methyl group that is low in 13C, like terrestrial carbon. And then we make this intermediate acetaldehyde. And then it has to react with this hydrogen cyanide that has a certain isotopic composition, a little bit heavier than typical terrestrial materials. And then it goes through this fractionation that we determine by experiment, and then you get the resulting structure. Many parts of this are subject to test, okay? So we know that there are reduced carbon pools in these meteorites. Most of the reduced carbon in the meteorites is like this. We know that it has acetaldehyde like this. We know the hydrogen cyanide is like this. We determined this in the laboratory. We measured this in the laboratory. We can create a self-consistent scenario with lots of different checkpoints that work. So next we did the same thing on a dicarboxylic amino acid. It looks a lot like alanine, except it has a second carboxyl group hanging off the end. And you'd be surprised how hard it is to come to an understanding of where that second carboxyl group comes from. And so we chose this specifically to open up another window on the types of chemistry that were possible. Sort of shockingly to me, it looks an awful lot like the alanine isotopic structure. This is a preliminary measurement. It was done by a slightly different technique. It's less precise, but the signals are so big that I think it's interpretable regardless. We see that the carboxyls at both ends are basically terrestrial. So the methylene, the CH2 group in the middle of aspartic acid, looks like the methyl group at the end of alanine, and the amine carbon again is super enriched. You don't have to be a genius to have the idea, I'll bet the chemistries were similar. I'll bet there were similar precursors, similar mechanisms that were involved in this chemistry. And so we've proposed the following, that we need to somehow add a low 13C carbon at the reduced end, at the methyl end of this aldehyde precursor. That's a difficult thing to do, and we think it occurred to make the nitrile happened in the interstellar medium because we see such compounds in the interstellar medium. And then we undergo the Strecker chemistry of cyanide reaction, hydrolysis, to create the carboxyl, turn that into a carboxyl group, and to create the carboxyl group at the other end. But the key thing is we have this precursor that's 13C rich, and it is carried through and ends up at the amine carbon site. Okay, next we turn to nucleobases, which are the first step in the chemistry towards DNA and RNA. And so with this, we explored through a chemistry that involves formamide. So it's basically ammonia and HCO make this crazy compound. And then that can react with cyanide through multiple steps. I think there's like 17 steps or something to make adenine, which is a simple nucleobase. We don't find adenine on the Murchison meteorite, but we do find a hydrolysis product of it, xanthine. And so it's a reasonable guess that xanthine is derived by hydrolysis of adenine. And so we can take our experimental study of the synthesis of adenine, compare it to the product xanthine, and then make a guess about what we think that precursor had to be like, given the isotope effects we observe and given the final product. And so what we see is this precursor, we need there to be a 13C rich precursor. In principle, it could have been that cyanide in this case, but we already have this separate constraint. It says the cyanide is low in 13C. And so a more plausible scenario is we have this CO rich, 13C rich precursor, and it needs to have the same enrichment that we see in the amino acids. So it's starting to feel like I can do multiple kinds of chemistry. I can do nucleobase chemistry, I can do amino acid chemistry using the same families of precursors. We haven't yet fully characterized the isotopic structure in the experiments, and we haven't been able to measure the isotopic structure of xanthine in the meteorite, but that's sort of on our to-do list and will help us refine the story even more. So the next thing we did is we said, okay, we have these several cases where we've really worked out how the isotopic structure and molecular average content relates to certain mechanisms, and we've started to develop a hypothesis. Let's see if we can now mine data out of the literature of the compound-specific isotopic compositions and build out that hypothesis to see how far it can, how many compounds can it encompass before it starts to break down. Here's one example. There's a lot of this sort of literature mining and hypothesis developing. I'll just show you a couple of simple examples so you understand how it works. So here we take a bunch of aldehydes that have been analyzed in the Murchison meteorite, and we say, what if the aldehydes all were made in the way that we said acetaldehyde was made? That is, we have a 13C rich precursor that's carbon monoxide, and then it joins with a reduced carbon pool that is 13C poor. And we'll just say it's exactly the same 13C contents that we required from acetaldehyde to explain our laboratory results for the amino acids. And here's what we observe is it will predict the little circles, okay? And then the data are the little squares. And if you know anything about meteorite compound-specific carbon isotope measurements, you'll think I either cheated or it's a miracle or something like that, that they're within 50 per mil of each other. But it's like it's not a bad first order match, except for the smallest compound. So formaldehyde is lower in 13, it's 13C rich, but it's not as 13C rich as we require for this hypothesis. We don't really know exactly what that means. You can just note it is in the right direction, but not perfect. Formaldehyde is pretty easy to lose and to contaminate. It's one of these volatile compounds that is easy to disturb. And so we're more trustful of the refractory compounds that didn't move around as much and aren't as present in our environment. Here's the same idea for the amines. We said, okay, what if we just, on the way towards making amino acids, you make amines? What if we said, okay, all the amines are just the byproducts, the side products of our amino acid synthesis? In that case, they should be like these circles, and the data are like these little squares. Not perfect, but it's the same pattern, it's the right order of magnitude, it's really doing pretty well when you consider the standards of the data and our knowledge of the kinetic isotope effects and so forth. So by this sort of line of reasoning, we have built up a reaction network. The actual reaction network, if I showed you the whole thing with all of its elementary steps, you would not be able to see anything that was going on. It would look like the London metro map, okay? There's many, many, many lines. This is a simplification of it. But it basically says there's a family of chemistries that happen. We'll talk about in a moment where they happened. Probably they're the interstellar medium. Simple chemistries that we see happening in interstellar space by spectroscopy, where carbon monoxide, the ubiquitous sort of precursor of carbon chemistry in the interstellar setting, is able to join with reduced carbon pools to make aldehydes. It's able to react with hydrogen to make formaldehyde. That's able to react with NH compounds to make formidine. Cyanide is around, ammonia is around, oxynitriles are around. These are hard to make in any normal lab sort of context, but do get made by photochemistry in the interstellar medium. So this whole realm here, we know occurs in something like this simplified form in the interstellar medium. And then those precursors can react with each other to go off and make these other things. So the oxynitriles can react with cyanide, with ammonia to make the dicarboxylic amino acids. The aldehydes react with ammonia, with hydrogen cyanide, with water to make the monocarboxylic amino acids. Oxidation of the aldehydes makes the monocarboxylic acids, and so forth. So we're building out this network, and this is just sort of a broad brush stroke. You won't be able to read it very easily in the time that I'm going to give you. Gives you a sense of the scope of comparison of data with models. So all of the gray blobs are predicted ranges in carbon isotope content for it's not even been tuned very much at this point, but a sort of initially proposed version of this reaction network. And then all the little dots are data. And in some cases, the prediction is a little teeny dot and the data is a little teeny dot. Sometimes we have to consider reaction yield, temperature, kinetic isotopic effect, size, things like that. And then our expectations blur over maybe a 10 per mil range, something like that. And then these are all the straight chain compounds. These are all the branched compounds. There's a couple dozen of them, maybe 3 dozen of them. And you can see exceptions. There are a couple of, some of like the C3 monocarboxylic acid, that doesn't look very good. We already talked about the formaldehyde, that didn't look very good. But by and large, it's a very successful description of the differences between groups of compounds and even within groups of compounds, their relationship to size. So we feel like it's a pretty good, it's not doing a bad job at this point. Okay, so now let's close by talking about interpretation. What do we think this means? What do we take away from the inferences from the existence of this reaction network? So first of all, the roots of it, like I already mentioned, are very similar to the chemistry of the interstellar medium. We have carbon monoxide is ubiquitous and abundant, ammonia, hydrogen cyanide. These are absolutely critical. Water, without these things, this chemistry cannot occur. We also see some of the immediate intermediates, okay, things that are formed from that first group of compounds, but then we also need present in abundance to then feed the prebiotic chemistry we care about, like the nitriles, like formaldehyde. Okay, we also note that we're aware that not only is there vapor phase chemistry that's sampled by rotational spectroscopy here, but we also know from the laboratory that there are ice grain reactions that take these precursors and cook them up even further into larger compounds. So it's plausible that we are polymerizing, oligomerizing, making bigger aldehydes and so forth that have not been seen in interstellar medium spectroscopy because they're not volatile enough. They're all in solids. Okay, we also know from looking at the spectroscopy of disks that some of the precursors that we need for abiotic aminosynthesis, like methylcyanide and so forth, also are seen in those settings. So not only the interstellar medium, but also disk chemistry could be contributing to some of the early stages of this. But we know by looking at the most primitive products of disk chemistry, here's mass spectra from the gases blown off from a comet in the solar system, we can see amino acids present in these materials, but only the simplest of them, like glycine, the smallest, simplest amino acid. So we know we're doing qualitatively the chemistry that needs to be done in objects that have not been part of planetary bodies or even significantly heated up asteroids. They're not even as much thermally mature as the carbonaceous chondrites, but there seems to be a limit. Okay, we're not seeing the full richness and the size range of things that were observed in Murchison meteorite and that we studied. So we feel like the most likely place to host most of this reaction network, that is, think of it as kind of like an octopus. Okay, the octopus has a head, and the head is almost certainly interstellar medium products and maybe disk products. But all the arms of the octopus are things that are happening together and drawing on those same precursors, and they all involve water and so forth. We need an environment for those. And the interiors of the asteroidal bodies that are the parents of the carbonaceous chondrites strike us as a good place to do this. So when I was last at Hebrew University, one of the other things I talked about was reconstructing the aqueous alteration, the temperature histories of these bodies. This is just the punchline from that they were, clement, they were moderate in temperature, water was present, they were carbonate saturated. That lets us make statements about the pH. We know they were relatively reduced, but just above the water-hydrogen buffer. So we can place them in an EH-pH diagram, and we know broadly the kinds of chemistry that should have been possible. And we know what kinds of solutes were present because we see ammonia, we see hydrogen cyanide and aldehydes. We think this was the sort of cooker that did this chemistry. We think we just did a dry run maybe of the chemistry of Enceladus. So it's known from mass spectra from the Cassini mission that the plumes of Enceladus are ejecting organic rich solutions that include some maybe aminated things, some nitrogen-bearing compounds, maybe we just studied the kind of chemistry happening there. This chemistry is also very similar to the cyanosulfidic hypothesis chemistry that was hypothesized to occur in lakes in the early Earth. So some of the details may be relevant for that. And then we're basically done. Let me just point out a few long-term goals. We're going to do this on material returned from Bennu if that succeeds. We also have a part of my group applying these concepts to the study of ancient organic matter. Basically, we would like to take kerogen, the oldest kerogen you can find in the earth, liberate moieties from it by hydrogen pyrolysis, and then study their isotopic structures and see, can we tell that these were originally biomass, or maybe they weren't, okay? And then Mars sample return. And of course, the Mars sample return opens up one of the more exciting possibilities. Maybe we'll find organic molecules that were made by life on another planet. How would you know? How would you use these concepts to comment on that possibility? First of all, we can use the specificity of isotopic structure to tell whether or not something is a terrestrial contaminant, because we can compare it with terrestrial examples. Now that we know the meteoritic isotopic fingerprints, we can falsify whether it's just meteoritic infall. We can figure out whether it matches any of the abiotic syntheses we've studied, like the Strecker synthesis. We can look for examples of idiosyncratic patterns of isotopic structure that imply the construction of things from little precursors in specific ways, like fatty acid synthesis. And we can build up by studying multiple compounds from return samples, their own network of reactions and see, does that look like a metabolism or not? And finally, we can use clumping concepts to assign temperatures of origin to some of the compounds and see whether they're consistent with habitability. Okay, that's it. I'm gonna stop sharing. How did I do? I did not so good. I actually did an hour, but then again, I don't know. I did my best. So.
You did well. Thank you so much, John. That was great. And I'm opening the chat to see if there are any guys. If you have any questions, please text me and or write it in the chat, and I will call you. And if not, let the shouting begin. Anyone? Okay, I'm sorry, there's construction going on in my wing of the building, and at some point the alarm went off. So there was a lot of action here. I have some questions. So can you? I think you implied that about the source. You basically show that there are two source materials that are very different isotopically. And if I understand right, and what I'm looking for is some clarification, they come from different part of the disk. Is that where you?
Yeah. So carbon monoxide harvests C13 out of the free carbon pool, the atomic ionic pools of carbon in the interstellar medium. And it does it for a relatively simple reason, that the CO bond is a strong potential energy well. Right. And so if a carbon-13 falls into that well, if it makes a CO bond, then it's much harder to dissociate and reliberate than if a C12 does. And so when you look up in the interstellar medium, at least in the local region of the Milky Way galaxy, and look at the C13 version and C12 version, you can see that the CO is enriched by like a couple hundred per mil compared to like the background of other carbon sources.
And at what point of I guess expansion and information of this do you think this fractionation occurred, or this signal was acquired?
So that signal is acquired during the photochemical processing of atoms and small molecules in the interstellar medium. So it's prior to the accretion of disks. Okay, once you take all the molecules and you bring them into an optically dense and optically thick cloud, Now they are shielded effectively from radiation. And so the chemistry that's occurring in the interstellar medium slows, and other kinds of chemistry, thermally activated chemistry, normal chemistry, takes over. And so these really strong enrichments, the origin of this 13C rich character in carbon monoxide compared to other carbon pools, it's a kind of fossil remnant of this diffuse state where a lot of photons are flying around and activating, driving ionization and reaction. And then there's a catalytic property to the ice grains as well that helps some of the chemistry, helps the carbon then. So what one response to the story that I've told you is people who are really focused on ice grain chemistry wonder whether all of the chemistry I explained to you could happen in ice grains in an interstellar medium, and that the meteorites just inherit all of those compounds. They don't cook them up there. I don't know how to falsify that yet. We don't know enough about the mechanisms and the isotope effects associated with that chemistry, sort of for logistical reasons. If I want to study what the Strecker chemistry is like in aqueous environments at 50 degrees C, well, this is trivial. I just go into a hood. Well, no, I ask somebody to go into a hood and spend 3 years or whatever, and then they come away with an answer. If you want to do the same thing for ice grains, well, you're going to be doing non-equilibrium chemistry. It's a very difficult experiment. You don't know what version you should do and whether it's really that similar to the ices you examine that are in nature. It's much harder to flesh out how that should operate. Thank you for your comment, Aldo. I'm very much looking forward. Oh no, he just wrote to me privately. Anyway, somebody said something nice to me and I'm saying something nice back. Thank you. Glennis, a colleague of mine from NYU, writes how much variability in initial 13C concentrations, maybe between CO in different settings or between CO and the organic pool, the reduced pool in different settings, should there be in different systems? This is something that's definitely a local fingerprint. Like if we had the version of the Murchison meteorite from Alpha Centauri, maybe that's too close, from the other side of the Milky Way, then it could have a radically different total carbon-13 pool, because that is a fingerprint of the weighted average of all of the nucleosynthetic history of the carbon that has reached the chemical environment where you're doing the synthesis that you're interested in. But then also the photochemistry, the nebular chemistry, the ISM chemistry that occurred prior to the accretion of solid bodies, that also would have had a different history and could have been radically different by hundreds of per mil. So really, we're talking here about the reconstruction of a local story. And if you applied the same principles to something taken from another galaxy, Andromeda or whatever, or the other side of the Milky Way, you expect to begin with fairly different starting points. There is a sort of predisposition to CO being 13C rich compared to reduced carbon. And the chemistry is the chemistry. If you create an aqueous body and it's 50 degrees C and there's cyanide, you're going to do this chemistry. But you may end up with different patterns of isotopic variability. Okay, let me see. I think that basically we have examined data for 5 other primitive solar system meteorites. And they're not identical, but they're surprisingly similar for the major themes. There's also a version of this story about deuterium, where we put together a story about how the deuterium enrichments that you see in these compounds originated. And that story is also pretty similar for different meteorites. But with the caveat that we know an enormous amount about the Murchison meteorite. Dozens of molecules have been analyzed for their compound-specific composition. We have site-specific structures for a few, whereas these other meteorites, one meteorite will have 5 compounds measured in it, and another meteorite, 3 other compounds. So it's just very patchy outside of the Murchison meteorite. Oded, I see you.
Yeah. Do you see any such effect in any terrestrial compound, like strong enrichment of one site?
Yeah, this is a good question. So we have a large body of work, a lot bigger than the body of work I showed you here, on catagenesis and natural hydrocarbons. And just for simple technological reasons, most of that work focused on the small gaseous products of thermal breakdown of organic matter, hot methane, ethane, propane, like that. And in those compounds, there are some big deuterium variations, but the C13 variations, they tend to be an order of magnitude smaller than what I showed you. An exception is a study of the methyl group from the methoxy moiety of lignin and of lignites and of coals. So coals have this ring-dominated structure, but the rings are decorated by methoxy groups, which is an oxygen with a methyl group on it. And those methoxy groups, they're relatively labile. Bugs like to eat them. If you thermally heat lignin or coal, it will break that down first. So they're easily removed. And if you measure the concentration of them as a function of the maturity of a coal, they drop by orders of magnitude. So in that material, the delta 13C of the methyl group can rise to plus 30 per mil delta 13C on the PDB scale, so 60 per mil above where it began. This isn't 150, but it's not nothing. It's really quite big. And this is because it's the residue of a Rayleigh distillation that has an elementary isotope effect that's typical, 10, 15, 20 per mil, like that. And so because the extent of reaction is so profound in the last residue, it gets driven up to very high 13C contents. Not quite what we see in the meteorites, but very high. So it is possible that you could create a reservoir in this way by a normal chemical process without this sort of inheritance that I've described. I would say, that the reaction network that we've proposed, it's not like some fact that we found lying on the ground. It is an elaborate chemically informed hypothesis, and it can break down. It can be disproven, and you can come up with an alternative hypothesis. But the trouble is, your alternative hypothesis has to be similarly complex, has to deal with a similar diversity of constraints, and somehow has to hang together as good or better. And so I haven't come up with a version of this where I create this 150 per mil signal by a normal chemical process, like a distillation. But it's possible, it's imaginable.
Thank you. Now, do we have any other questions, comments? Right.
I asked one last question. Fingerprinting of life. Do you have any thoughts about a way to do this? Some insights from all the modeling you guys have done? Something that you would see in a simple molecule that would tell you this?
So I think there will not be a single fingerprint of any given molecule that says you must be life. Instead, my expectation is that if you take a new sample and it's important enough to you, you care enough that you will study it as carefully as we've studied Murchison, then you will be able to build up a network that is based on inductive reasoning, is based on tracing isotopic fingerprints. This one begat that one, like that. And that when you are done building that network, you will look at it and say, that's a metabolism. And so one example of a kind of fingerprint we know about from terrestrial life that would be a clue, you'd say, wow, that really seems like a metabolism to me, is the fatty acid isotopic structure. So fatty acids are chemically simple. They're just linear hydrocarbons with a carboxylic acid at the end. Boring. Lots of ways to make them. But when life makes them, it builds them out of Legos that are 2 carbons big, the acetyl group. And the acetyl group has a heavy end and a light end because of the way it was made from its C3 precursor. And it's a pretty big difference, 10, 20 per mil difference. And when you build the fatty acid, you take the Legos and you attach them head to tail, and it creates a zigzag carbon isotope pattern like Charlie Brown's shirt, okay? And that is something, maybe you can come up with some kind of catalytic scheme that's non-biological that will take a C2 group, fractionate it, assemble it in the correct way head to tail, even though in the end they're the same chemical setting. You know, I'm going CH2, 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 yet they remember that one of the CH2s used to be a methyl, while the other CH2 used to be a CO something bounded to an enzyme. They remember this. And so these kinds of patterns, I think, if we find the products of metabolism on Mars or Enceladus or whatever, we will see patterns like this and we'll look at them and say, you know, this is crazy. This did not get made by Strecker chemistry. This did not get made by polymerization of alkanes or whatever.
Okay, I see Oded has one last final question.
Yeah, shoot, I can go all day.
I know.
Chirality is strongly connected to life.
Yes.
What does chirality?
Falsely, but strongly. Yeah, go on.
Okay. I'm not an expert in this field, but
Yeah.
Does chirality has an isotopic effect?
Yeah, this is a good question. You can turn it around and ask, do isotopes have a chirality effect? Okay, that is known to be true. So chirality can be manifested without life. Okay, you need a selection rule in non-biological systems that selects for, first of all, you need to be making a selection. You need to be separating. And that can be, say, by condensation, growing a crystal, an organic crystal is one way you could select. You can select other ways, absorption, evaporation, whatever. And then if you have even a slight energetic difference between chiral forms in solubility or whatever, then, and you garden, okay, then you can migrate from almost no preference to a strong preference and grow a chirally pure crystal. We know from experiment that if you isotopically label the chiral forms to be different in isotopic content, that has a similar effect. The vibrational isotope effect on the energy of the molecule is similar to the chiral effect and similar to the selection effect that's required. So I can force a chirality in a crystal I grow or something that I absorb by isotopic labeling. In the case of experiments, you do this with 100% labels and you create 100% pure molecules. But you should expect that this also will manifest as isotope effects in just non-biological processing. But the trick with chirality is, it's true that terrestrial life is strongly chiral in its preference, at least for some of its structures. Not all of them, it's for some of them. And that it has to be basically enantiomerically pure for those functions. But there are inorganic ways to be strongly chirally selective. And there are chiral compounds in the Murchison meteorite that are not enantiomeric, they're not racemic. They have enantiomeric excesses. And it's when you go into environmental samples and look for biomass using enantiomeric excess, they're typically subtle. It's not like pure, because racemization, interconversion of the enantiomers, that occurs spontaneously as well. So it's really, in practice, it's shades of gray. It's not really like a stark signature, biosignature.
Okay, thank you so much. I think we're good with questions. We don't have any more questions from the audience. So now you can imagine a roaring round of applause.
I do it for myself. I imagine that all the time. That's how I go through my day.
All right.
All right. Thanks for the invitation. It was great to see you all again. See you all soon in person, I hope.
Okay. Thank you so much, John. Bye. Okay, I'm going to close the meeting.